Okay, good afternoon, everyone. Welcome to today's installment of the 2021 COVI Lecture Series. Thanks so much for joining us. My name is Sarah Ackles, and I'm the Marketing and Communications Officer at Brock's Cool Climate, Enology, and Viticulture Institute. I'll be moderating today's lecture for you. Even though we're completely online this year, we're still going to be doing our typical question and answer period. So I encourage you to please put your questions into the chat feature. And at the end of the presentation, I'll ask some of our speaker. Today, I'm very pleased to welcome Dr. Sudarsana Poojari, who will be our speaker this afternoon. Sud's a senior research scientist here at COVI, where he specializes in grapevine virology, as well as holding adjunct professor status in Brock University's Department of Biological Sciences. Sud earned his PhD in plant pathology from Washington State University back in 2013, and he completed his NCERC postdoctoral fellowship at Agriculture and Agri-Food Canada at the Summerland Research and Development Center in British Columbia. While he was out in the Okanagan Valley doing that, his work focused on understanding the epidemiology of grapevine virus diseases. Sud has continued to research advanced molecular diagnostics and the epidemiology of grapevine virus diseases while here at COVI. He also leads the National Virus Testing Facility for Grapevines, as well as leading the academic team of scientists on the Clean Plant Extraction Sequencing Diagnostics Initiative, also known as CLEANSED, with partners across the country. Today, he'll draw from that wealth of knowledge and experience and speak to us about advances in grapevine certification standards. So with that, I'll turn the floor over to you, Dr. Poojari, and please go ahead and give your lecture.
Okay, thank you, Sarah. I'm going to share the slides now. All right, just a minute. Can you see the slide, Sarah? Hello?
Hi, Sud.
Can you see the slide?
We can, yep, you're all set to go.
Okay, perfect. Thank you very much, and good afternoon, everybody. And today I'm going to talk about the advances in grapevine certification standards. That's the outline of my presentation today. One thing that I realized while researching on this topic over the last few days, that grapevine certification, talking about grapevine certification standard is not, it cannot be covered in one talk. Maybe I should follow with another session on this, but I try to be reasonable and try to cover as many things as possible. So whenever we talk about a certification, we talk about certification for what? Especially when it comes to grapevines, we are talking about certifications for grapevine viruses as well as other pathogens, just like in any other crop commodity. Grapevines are not different in that sense, but one of the things that we need to be knowing before we understand what is the certification and also what is the clean plant programs are all about, it is very important to understand what type of grapevine viruses are there and what is their importance in particular region, for example, and how to detect them. And then I'll be talking about the certification standard, what is the structure in different scenarios, as well as I'll be talking about the clean plant programs, how different they are in different countries and why. And also at the end, I will be talking a few slides on the Canadian perspective on the grapevine certification network. So the first is the grapevine viruses and their importance and detection. So the first thing that we have to see when we talk about the grapevine viruses is how many viruses that grapevines are susceptible to. As you can see in this diagram here, which shows if there are any cross or mixed infections with other fruit trees, example, for palm trees, apples and pears, and stone fruits, cherries and plums and the nectarines. If you see this diagram, it is very clear that the grapevines are unique in the sense that there are majority of viruses that are very unique to grapevines, not co-infecting other palm trees except few viruses here. But when you look very closely, what are those, what do they do? Although there are more than 80 plus different virus species that's known to infect these grapevines, but not all of them are important in terms of how they cause significant negative effects, especially in terms of affecting either it is rooting ability or grafting intake or vine vigor or fruit quality. So some of the major grapevine virus diseases I have outlined here. When I say diseases, they may be complex, caused by either one or more than one species of viruses. For example, as you can see, grapevine leaf roll disease is caused by a complex of viruses called grapevine leaf roll associated viruses, numbered from 1 to 3, 4, and 7, as well as grapevine associated virus 2. And then grapevine red blotch virus, which is relatively new virus species that we have known so far. And there is a rugose wood complex, again, caused by many vitiviruses like grapevine virus A, B, D, as well as the rupestris stem pitting associated virus. And then there is a grapevine fan leaf degeneration complex caused by grapevine fan leaf virus. There are other viruses as well called grapevine fleck virus and grapevine pinot gris virus. But the thing is, when you talk about different viruses, it is very important to know which of these viruses are prevalent in one particular region, especially when you are trying to design a certification program for a particular country or region. Okay, so the question is, why do we need certifications? As you all know, the grapevine planting, grapevines are propagated by the cuttings, the planting material, which is vegetatively propagated plant. So whether in Canada we use grafted vines. So it is obvious that if it is a rootstock that is infected with the virus or the scion material, the resultant daughter vine is going to get the virus because the virus can move from infected rootstock to healthy scion or a healthy scion to infected rootstocks. In case of own rooting structure, if you take cuttings from an infected mother vines, obviously the daughter vines are going to get those virus. So there is no way that we can cure the virus once the vine is infected. So one of the best things that we can do is to make sure when you are starting a vineyard to plant the vines that are clean of viruses or free of pathogens as much as possible. And that's why the certification comes very effective. So the importance of testing or detection in certification standards. Why? So if you look into epidemiology of these different grapevine virus diseases and how the biology of these viruses are in the grapevines, for example, not all of these viruses show apparent symptoms, especially in hybrids. Some of these viruses they stay latent, and also it is important to know in a certification standard if you are detecting a virus in a particular vine coming from which source. That kind of gives an advantage to eliminate the source of infected material. And also in terms of plant protection and quarantining, it is very important to make sure that you have a reliable as well as sensitive diagnostic methods. And obviously, diagnostics plays a big role in disease management. And finally, the sustainability of grapevine production system depends heavily on the health status of the grapevine planting material, and it is critical that when you're starting, it should be free from grapevine pathogens. So that being said, what are the options that we have in terms of grapevine virus diagnostics, especially if you want to use them in certification standards? The first thing is symptom-based. The monitoring based on the symptoms is one of the methods that the grapevine certification standards recommends, but at the same time, there are so many anomalies so many inaccuracies in terms of detecting these viruses based on symptoms. As I just mentioned, there is possibility that these viruses stay latent without showing any apparent symptoms, and some of these symptoms mimics with other virus species, so you cannot really differentiate which virus it is. And also there is a possibility of confusing these virus symptoms with, for example, mineral deficiency. And the second method is the biological indexing. So what does that mean? So basically, this is a very standard approach that is used in the certification standards, where if you have an infected or a planting material that is suspected for a virus disease, and you take that material and you either graft the healthy material on top of those material and see if the symptoms, if you can reproduce on the healthy part of the material. So obviously this requires a little bit of training and experience to do this kind of biological indexing, as well as, it is obviously time sensitive and time consuming and labor intensive. And especially when you are looking into the perennial crops like grapevines, the appearance of symptoms might take a lot of time. That's one of the major limitations with the biological indexing, but it's one of the standard practice that many grapevine certification standards will follow. In the serological, especially ELISA enzyme-linked immunoassay, very commonly nowadays known as antibody test, very specific, can be used on a large scale. One of the limitations with this particular method is antibodies that is required to detect this particular antigens on the viruses are not available for all grapevine virus species. And it is considered as very less sensitive than the molecular techniques that are available today. When you talk about the molecular techniques, it is either polymerase chain reaction, standard endpoint polymerase chain reaction, or quantitative polymerase chain reaction, or we have different methods in the quantitative PCR, like digital droplet PCR, which is an absolute quantification PCR, which can tell you not only just whether the virus is there or not, but also tell you the concentration or the titer of the virus. And more recently, NGS, or next generation sequencing, or more commonly known as high throughput sequencing, is also being used in diagnostics of grapevine viruses, which is highly sensitive, and you can detect multiple viruses in a single assay. With all these options available, now we're going to see these different certification standards, what their recommendations, how they are recommending these different methods, how they're using this method for detecting different viruses. So that takes us to the next topic, the certification standards, the structure. So if you look at any certification standard, there are 5 different stages. The first is the selection of varieties or rootstock of different grapevine planting material. So here, you cannot really depend on the symptoms. Obviously, when you are selecting, you can monitor for the virus-like symptoms, and you can use the serological molecular test to confirm if there is any viruses are present or absent. If there is any virus present, there is an elimination process in the certification programs. I will be talking a few slides on the elimination process. And the second is the production of the material called nuclear stock, where when you have a planting material that is tested negative for certain number of viruses in your certification standard, you can consider that as a candidate for the nuclear stock. So where you can start propagating the material from. And this propagation can be done if the material tested negative, you can then by traditional methods, as well as you can go and do a meristem tissue culture or in combination with the treatments like with the heat treatment. And then comes the how do you maintain this nuclear stock using either the best management practices that are recommended by regional or national agencies, regulatory agencies, and also some of these standards regularly require you to retest those nuclear tests to make sure that this material is free of any particular pathogens. And then the propagation block. You take any material from this nuclear stock and start establishing a propagation block where you can always follow, you have to follow the best management practices to make sure it is free of any pests. And then it comes to the certification, certified plants, where you take material from the propagation block and start propagating them or grafting them or using them as a conrooted. And then you have to follow the standards of this propagation block, even in the certified blocks. That's a very typical structure of a certification program. But again, put that in a graphical perspective. Again, my apologies. I just made these graphics with the help of my 8-year-old. So, I probably have much more time these days. So again, the 5 stages to put into perspective, as I shown in these pictures here, the cyan and rootstocks in different colors. If it is tested negative, it will become a candidate material, and then it will become a nuclear stock, both cyan varieties and rootstock varieties, where you propagate that and then call it as a propagation stock. Either this can be grafted just as a cyan variety or rootstock variety, which again, you put from here and go and use it as a certified block when you graft them. So that being said, especially when you look into the EPPO, European and Mediterranean Plant Protection Organization, excuse me, they have this Grapevine certification standards. Especially why I'm focusing on this is the Canada especially imports majority of the grapevine planting material from European countries, especially from France and Germany. So when you look into their certification scheme, again, this particular screenshot is downloaded very recently, and it is EPPO bulletin that is published in 2018. One of the things that I'll be talking a little bit more details in the next few slides, but one of the things that I wanted to highlight is how outdated these particular guidelines are. As you can see here, as I highlighted, it is first approved in 1993 and then revised in 2008, and now we are in 2021. So there is a huge gap between that update and where we are now, especially in terms of our advancements in understanding the epidemiology or advancements in disease diagnostics, especially in emerging new virus diseases. For example, grapevine Pinot Gris virus that is reported in Europe very frequently nowadays, and as well as the grapevine red blotch virus, which is a major concern, especially in the North American countries. So the EPPO guidelines on virus testing. Again, I'm going back to my basic structure of what are different type of testings that they're recommending. First is the biological indexing, where you test any pathogenicity on indicator plants, and it is a mandatory step that they're recommending from the EPPO, especially for leaf roll as well as the rugose wood complex. And the recommendation is always with the 3 replicates with a minimum of 6 to 8 grafts each, and that can be different type of grafting is recommended, whether it is a green grafting or wedge grafting or strip grafting. But more often, it is recommended that the green grafting would work with better efficiency than other 2. And then when it comes to testing, both serological testing as well as the molecular testing is highly recommended. When it comes to serological testing, they're recommended for grapevine fan leaf as well as other NIPO viruses and leaf roll viruses. There are some witty viruses, and there is grapevine fleck virus. One of the things that they're recommending is use the serological methods for detection of these viruses as a complement, but not as a substitute to other diagnostic method. That means whether it is a biological indexing or molecular testing. As for the same reason that the antibodies are not available for all virus species, and it is less sensitive than the molecular testings. Again, they also recommend the molecular testings for the viruses that are mentioned just above in the ELISA testing, and also they're recommending that it's very high specific. So the viruses, again, to go into more details on the EPPO guidelines, which viruses they do cover, is the grapevine degeneration complex. The number of virus species I listed here, I'm not going to read them out. And also there is grapevine leaf roll complex, grapevine rugose wood complex, grapevine fleck disease, and there are a couple of phytoplasmas also included in the list. But that being said, this is the EPPO guidelines. When you look into details of specific country guidelines, they do have additional viruses that are listed, but they vary in terms of how these standards in each country in the European region. One of the things that I wanted to highlight again, because this is an outdated certification scheme, the red blotch is not in the list. That is something that we have to take a notice, especially in the Canadian perspective. And again, going back to the EPPO guidelines on virus sanitation or elimination, the heat treatment they recommend to, in combination with the meristem shoot tip culture, is they say heat treatment has a variable levels of efficiency, and it is recommended for only cuttings that are 2 years or older, and the temperatures are listed there. And when it comes to the meristem shoot tip tissue culture, is one of the method to highly recommended method to eliminate plant viruses in many cropping system, and grapevines are not different. The recommendation to use the meristem tip, because it is believed that these cells are metabolically active and not having formed any vascular tissue in them. It is believed that the viruses will not be present in them, and that's why we have high rate of success if we use these tips 0.4 to 0.6 millimeters in size within the meristematic dome. We can generate the many plants that are virus free, and these meristematic tissues are grown in an artificial media until they grow into the size where we can move them into a normal growing media. And once you generate these plants, again, the EPPO guidelines suggest that you test as recommended for the number of viruses. So that brings into the next topic, the clean plant programs, how different they are and why. So before we go into those details, I would like to emphasize the fact that what are the factors that we need to take into consideration when you are designing a certification program that can fit into a national clean plant program. So one of the things is you need to be looking, especially when you're looking at the grapevines in your region or in your country, so which viruses that are important. So surveys, using different approaches for multiple viruses would definitely help, and understanding the epidemiology of those viruses will also help in terms of, is there any viruses that are mild or it doesn't cause any significant losses in terms of production as well as the vine growth. Can it be eliminated in the certification program, or can it be tolerated, excuse me. And also, is there any possibility based on your local or regional surveys or national surveys, is there any region in particular area you can eradicate a particular virus species? And how to adapt different diagnostic methods that are developing as we speak every day. There's new diagnostic development coming up. How to adapt these diagnostic methods in terms of certification as well as the regulatory aspects are concerned. And once we have the clean plant program established, how to protect it, how to prevent the secondary infection is also part of the clean plant programs. And some of the clean plant programs I will be talking, I think they have witnessed the success stories of their clean plant programs as well. So with that, I would like to present the basic structure when you look into the clean plant programs for the grapevines. So especially when it is more relevant to the Canadian perspective, because here in Canada we divide this generation. We have this generation level concept that is used to define the planting material or categories into the clean plant programs, and also signifies the degree of which this plant stock is related to the originally tested material. For example, here, generation 1, G stands for generation, is the mother vines that are selected and screened for known pathogens, again using different type of testing approaches. And typically these blocks called either foundation blocks for example, in the States they're called foundation blocks or nuclear blocks, or just as a G1 material in Canadian perspective. And then these blocks are maintained in isolated areas or in the screen house or polyhouses. These are the sole source for the G2 planting material. So that applies for the next generations. G2, G3 comes from G2, G4 comes from G3. And then as you see, one of the things that I would like to highlight that the grapevine planting material, for example, G2, it should be propagated from G1, not from the secondary source. And also sometimes it is very confusing as for example, different nurseries sourcing G1 material, and when it comes to G2, they should not be exchanging materials in between them. And that's one of the things that's very important when you are designing the clean plant network. Make sure that those guidelines are strictly followed. And then the G3 material definitely comes from the G2, and sometimes they simply call as increased blocks in some areas. And then G4 is typically what we call certified blocks, but again, it depends on the size of the industry, size of the nurseries, whether they would go for this G3 or simply they sell G3 as a certified block. So when you look into the clean plant network in the States, for example, it's called National Clean Plant Network. Although they have this national clean plant network that is a collaborative approach between different universities, the federal and the state government organizations, the regulatory agencies, especially it is linked with the USDA's Animal and Plant Health Inspection Service, or called APHIS, which is very equivalent to Canadian Food Inspection Agency here in Canada. It is mainly state controlled, these certification programs. For example, as I mentioned here, the California Department of Food and Agriculture, CDFAs, have this grapevine registration certification program, which is one of the oldest certification programs around the world. It is established in 1956. I think it's followed by Cornell in New York. I think it is established in somewhere in 1960s. But they do have other centers, for example, in Washington, Missouri, and Florida, that these, especially for focusing on the grapevines. So how do the structure looks like? Especially when it comes to clean plant network in the States, it's a foundation plant services in a situation in UC Davis that has this registered foundation block. And the recommendations that they do have to use, well, they come up with a protocol called the Protocol 2010, which is nationally accepted protocol in all different certification standards. And the basic structure is, as I mentioned before, they have a foundation blocks at FPS, and where the nurseries will collect the material from and have their increased blocks and certified nurseries plantings. And from there, it goes to the food production or vineyards, the commercial vineyards, or to the growers. This link is very important to maintain, to make sure that they follow this protocol 2010 at the beginning, and make sure that in different stages of their certification, certain testings and certain monitoring is taking place. And then again, to reiterate that the Foundation Plant Services is a service department in the College of Agricultural and Environmental Sciences in the University of California, Davis. And this department performs the disease testing that is required by the CFDA's RNC program, and maintains the foundation vineyards that supplies nursery material to the participants. And as you can see, the recommendations for the virus testings include serological, biological indexing, as well as the PCR methods. They do test for 30 plus viruses, as well as the phytoplasma and Pierce disease. One of the recent advancements in their certification standards is the use of high-throughput sequencing as a tool for fast-tracking this process. And this is still in the preliminary stage. I think there are a lot of collaborations going on to harmonize this, to use this high-throughput sequencing to be verified and used in different programs. But once it is tested with HTS, I think right now in California, they do call it as a provisional quarantine release, which means that it can go to the nurseries, but at the same time they do test with the regular, the PCR as well as the biological indexing at the same time. So that makes into the next topic, what we have here in Canada. So there are two different things, the quarantine regulations as well as domestic certifications are two different things here in Canada. Canada regulates the imports of any grapevine planting material that is coming from foreign countries through CFIA. And Canada allows the import of this grapevine material that is certified under the United States certification program that is in California, Oregon, and Washington. Other than the USA, there are only, I think, 2 countries that are approved, CFIA approved, with having nurseries certified to export the grapevine material to Canada, that is France and Germany. So what CFIA does, if any new material that is coming to the Canada or any material that is being imported, is to look specifically into the regulated viruses. The number of regulated viruses from the CFIA is listed here. But as you can see, majority of the viruses that we do face problems in terms of significant economic damages is, for example, grapevine red blotch or leaf roll. They're not listed here because we do have those viruses in the country, and that's why they're not regulated. So that brings to the domestic certification standard. The Canadian Grapevine Certification Network, CGCN-RCCY, has started in 2017, and their first interim verification standard has came into effect in 2018, I believe. And they do have 2 certification standards at the moment. One is the interim verification standard, and the second is the long-term certification standard. So why 2? Because we started a little late, and there is an immediate need for the growers to source clean material, at least for the major viruses that we do have, with the knowledge that we have with our large-scale surveys, that we have established some certain viruses that are present widely or prevalently in Canada, in Canadian viticultural regions than others. So for that, interim verification standards especially checks for the 4 major viruses that are leaf roll 1 and leaf roll 3, grapevine red blotch virus, and grapevine Pinot Gris virus. So when you look into the structure of it, there are 3 stages. If any, Let me first tell you that this verification standard is available not only for nurseries, but also custom propagators, any growers or any wineries that are interested in propagating their own material, and if they want to involved with the verification standards, I think there is an opportunity for them to contact CGCN and initiate the process. When look into the stages, when a particular nursery or grower is interested in a particular block, the CGCN standards says that we'll first do the initial screening at 10% on a randomized sampling strategy. If it finds less than 15% virus infection for any of these 4 viruses, it will go to the stage 2 where the 100% testing will take place. And if it doesn't, then it will be dropped from the program. And then the second stage, when you do the 100% testing, if it is less than 0.1%, that is 1 in 1,000, it goes to the next step. But if it tests more than 0.1%, or whatever the viruses that are tested less than 0.1%, it will be subject to either removal of those wines or subjected to individual wine testing, because here the testing takes place as a 5-wine panel testing, that is composite sampling, just to reduce the number of tests and the amount of cost that is involved. And then at the stage 2, when the verification is complete, there will be annual inspection, and there will be a 10% random testing from that year onwards, just to make sure that they stay virus-free. How it works. The grape nursery, as I mentioned, the winery need to have to sign a contract with CGCN, and the samples will be collected and GPS mapped and tested for viruses at a CGCN approved lab. And one of the important thing is, which is very unique to Canada, I haven't seen this happen in any other countries, is the virus testing cost is shared at a 50-50 cost share between the growers and the nurseries and the CGCN. The nursery remits a wine per levy wine to CGCN on each wine sold, and the list of viruses, as I mentioned, or 4 viruses are listed here again. So coming back to the second, or the long-term certification standard, is mostly derived from the Canadian CFIA's export or import certification program. As you can see the structure here, there is a new varietal selection or anything that has to be gone through the certification program. It has to be tested first for 30 plus viruses. I have the list in the next slide. When it is test negative, it will go to the propagation and also tested for targeted pathogens again. And if there is any virus that is present, it will go back to the virus elimination process. Again, that includes same as heat therapy and tissue culture, mainly happens at the CFIA facilities at Sydney. And then follows the same pattern, the G1A or G2 or G3, G4, where in each, especially at G2 and G3, they will be monitoring, they will be testing for at least major viruses, if not all, using molecular testing approaches. These are the number of viruses that are covered under the CGCN long-term certification programs. As you can see, the number of viruses listed here, including the major viruses that we have, leaf roll 3 and the red blotch virus, especially covering the G1 and the G2 and G3. And also, because we are in the cool climate conditions, we are fairly bit susceptible to the crown gall caused by the bacterium, Agrobacterium-itis, than when you compare to some of the regions like California, where it doesn't express as much. Because of the cold winters that we have, there is a cold injury, it expresses more. I think that's one of the reasons that this long-term certification standards from CGCN is included crown gall in the certification standard. So what is the recent advancements? As you can see, I acknowledge this slide is a little hectic, but when you look at the left side here, it's a traditional approach where you have diagnostic tests for 30 different pathogens using different approaches. When you test positive a certain planting material, it goes to the elimination process, which is the meristem tissue culture. Takes maybe close to a year, and then the biological indexing it might take another 2 years, and then testing for these 30 different pathogens might take from weeks to months. But what we are working on right now, of course, this is a new approach, as Sarah mentioned at the beginning of my introduction, that myself and Dr. Mike Roth at CFIA in Sanitation, we're working on a new project called CLEANSED, using advanced diagnostic technique called high throughput sequencing, or basically what we are trying to do is to utilize this tool, make sure that it will fit into our program in terms of validation, in terms of how, in terms of reducing the cost or increasing the sensitivity. And also the time that is required for the whole process of coming back to the certified planting material, to reduce that whole timeline process by 2 years using different bioinformatic tools. And one of the major limitations when you're using HTS. And what I'm saying is HTS is not a foolproof method. There are still some biological characteristics that needs to be addressed, as well as the bioinformatic analysis that needs to be addressed. That's the whole project that we are working on. And this project is supported by Genome Canada. And I left a little link here to know more about this particular project. So that being said, I have lost two slides graciously provided by Mark Fuchs. What is certification? Certification is always a work in progress. For example, in the US or in Canada, all these certification programs are voluntary, and there needs to be a lot of work to be done on harmonization of these certification standards between different countries. Well, what do I mean by that? Excuse me. For example, in Europe, they do make sure that the clonal selection, the clonal identification, as well as the sanitary certifications are mandatory. But not many certification programs follow that. The country's directives in EU, European Union, are stricter than EU directives, for example. For example, on that is countries like Portugal, France, and Italy requires all the virus tests that is performed should be negative for Rugose wood complex, which is not required in Germany. Another example would be, for example, Spain would only ask for tests related to rupestris tempeting and corky bog disease. So those kind of differences are there. So when it comes to Australia and New Zealand, the standards include viruses like rupestris tempeting associated virus. What I'm saying is some of this certification programs do really eliminate this, do not include this rupestris tempeting virus in the certification programs because of the fact that it is widely spread and it is very hard to eliminate. And there is very little or no evidence of it causing any agronomically important or agronomically significant losses in grapevines. And in New Zealand, for example, if you see the grapevine standard version 3.1, which is published in 2017, they only test for LEAF-03. And in South Africa, on the other hand, they do recommend certification for virus diseases instead of virus species. And also they include the bacteria and other fungal diseases that cause grapevine trunk diseases, for example. So what are the challenges and opportunities? In any certification program, as I said, it has to be continuous monitoring, revisit the health status of wine in the foundation or G1 blocks or nuclear stocks, or if you're creating a new foundation blocks, it is always recommended to revisit the health status, monitor the test, monitor and test them to make sure that at regular intervals, to make sure that they're free of these viruses. And revise the certification standard programs based on surveys or epidemiological research that is involved, or any advances in the virus detection methods. And it's very important that multidisciplinary efforts are needed to maintain, remain strategically and be vigilant, and always look forward. And I would like to end with a statement that, when I was reading through different certification standards, most of the international regulations are encouraging to fast track the trade, but they are really limited or outdated in terms of updating their standards. For the successful clean plant program in any conditions, a continuous dialogue between scientists, grape growers, or regulators and the nurseries are very, very critical to the success of any clean plant program. And with that, I would like to thank all the funders and all the collaborators that I have. And I would give back the dais to Sarah for answering any questions.
Great,
Sarah?
Thanks so much. Yeah, thanks so much, Sid. That was a great presentation. Appreciate your time today, giving us all that great input. We have a couple of questions here, both of which are from Tim Martinson. The first of which is, how can 15% infection still be considered clean, he asks.
A good question, Martin. If you look very clearly what I mentioned in my slide on the interim certification standard is to, if you want to get into the program, we don't want to go and test 100% and put a burden on the nursery or a grower on the 100% testing. So that's initial 15% is to make sure, it depends on how many vines there are, and we are doing composite sampling. So we decided that, at least we test 10% and make sure that in that 10%, there is less than 15%, we'll go, and there is a choice for the grower or a nursery whether to go forward or no. So we need to give that option to the growers, to the clients, to make sure that, if they want to spend more money and go forward, we have to have that option before. If the grower decides, okay, there is a 15%, not even less, not even more, and there is an option for the grower to decide to go forward or not.
Great, and Tim, if you have a follow-up to that, feel free to just type it in the chat and I'll follow up, but I'll ask your other question in the meantime. So Tim also would like to know, for G3, G4, what will the testing frequency be? New York is striving for each vine tested on a 4-year schedule.
Yes, that's definitely on the long-term standard from the CGCN. There are two different things, there's G3 and G4. G4 under the CGCN program doesn't require to test anything, because that is the material that is going to the grower's field. So I think in majority of the certification standards, they're not testing the G4 material. But when it comes to the G2 and G3, there are different standards. Again, when you look to California program and New York program, they do test for certain viruses, major viruses, every 4 years. Either it is 20% every 5 years or 25% every 4 years. But under the CGCN, what we have here is the first year of those plantings, if it is the G3 block, doesn't need any testing because it's still the establishing block. And this, from the second to 5th year, it is with 20% for certain viruses. Again, we're not testing all the viruses. just to reduce the cost again, to make sure we're not putting too much burden on the growers or the nurseries. So it is at 20% for the next 5 years, so that you will cover the 100% within 5 years.
Okay, and Tim's actually sent a comment back, and he says, thanks for the answer, and that we do intense testing with the G2.
Sorry?
Tim says that he does intense testing with the G2 on their end.
Intend to test, you mean?
Intense testing.
Intense, oh. Again, it is good to test more always, but again, we have to balance things in terms of how the nurseries or the propagators will take that testing. But again, I have to reiterate that there are standard practices that CGCN has laid out when you are maintaining G2 or G3 blocks using best management practices, regular monitoring, and regular random testing. I think those things makes very effective in terms of managing those virus free. But as you said, if you are intend to test more, it is always encouraging.
Okay, great. And I'll just leave the line open for a little bit. So if anyone else has any other questions, please feel free to type them in. In the meantime, Sud, maybe I'll leave this opportunity for you to kind of share with everyone listening today if they're looking to contact you or to learn more about the testing or the work you're doing, whether it's from a nursery perspective or a grower perspective, what would be the best bet for them to do, the first course of action for them?
Well, definitely, if there are commercial growers who just want to test a few vines that are suspecting for virus diseases, the COVI virus testing lab is established with support from the industry as well as from the Brock University. So you are free to go and submit the samples. But if you would like to include or register under the either interim certification program or long-term certification program under CGCN, I would recommend you to contact Darian Temple at CGCN. She is the project manager she will have all the information that you need. And definitely I have put some links for the CGCN website there. There is information on how to be involved with the CGCN, and there are other educational resources as well. And definitely I put my contact information, email, phone number as well. If you have any questions, please always feel free to contact.
Okay, great. Tim said he had to leave, but he appreciates the efforts of yourself and your colleagues and all of the other researchers involved in this work. Very important work, of course, for our industry. If there's anything, Sud, that you would say looking ahead is kind of really exciting that's coming up in the work that you're doing, just to kind of give everybody a little preview of the work that is to come while we finish off here.
Sure. The couple of things I would like to say is this is detection. Some people have doubts on what is the certification is all about. Because one of the things that growers think that everything planting material coming from France, for example, Canada imports majority of their planting material from France, but they are limited by their own standards. And as a regulatory agency, CFI is limited by their own regulatory aspect of things. But that's one of the reasons that we need a domestic certification network, because in the domestic certification network, we will be looking into the viruses that are important to us, not in terms of regulatory perspective or some other country's perspective. For example, France never reported, or I don't think it is they are testing for grapevine red blotch virus, which is a major problem for us. So some of those things we should be taking into consideration. And that's one of the reasons that you should be asking, if you're buying a planting material, make sure it is tested for viruses like grapevine red blotch virus. And diagnostic methods, the second thing I would like to say is diagnostic methods is always changing. And one of the reasons that we are involving with certification agencies, regulatory bodies, and other academic institutions in terms of research collaborations on grapevine virus diseases is to make sure that we will bring that recent advancements in technologies in virus detection and make sure that we use that in our regular testing process or for research and development process, as well as the grapevine certification program.
Okay, great. Well, it looks like I think all of the questions for today, unless there's anything else that you wanted to add to the talk before we finished up here, Sud.
I think I'm good. If there is any more questions, I would be happy to answer. But if not,
okay, yeah, I think that looks like everything for today. And just so that everyone knows, we post this lecture up live on our website at brocu.ca/covey/outreach/lecture. You can watch this as well as the slides. There'll also be contact information on our website for Dr. Poojari, and you can contact him there if you have any further questions. But I'd just like to thank you for your time today. It was a great lecture. We enjoyed our afternoon learning more about the work that you're doing, the important work you're doing for our industry. And I'd like to let everyone know that our speaker next week is Jose Ramon Urias Torres, who is a research scientist at Agriculture and Agri-Food Canada's Pacific Agri-Food Research Centre, also known as PARC, P-A-R-C. He will discuss how to understand and manage grapevine trunk diseases with us beginning at 3 p.m. on Wednesday, February 3rd. More info can be found on our website about his talk as well. So thanks very much, everyone, for your time today, and have a great afternoon. Hope to see you next week. Take care.
All right. Thank you. Bye-bye.